Hi everybody, and in this installment we're going to be looking at the original paper discussing the GAPTrap expression system, and we're going to do that using benchling. So if you haven't already, I highly recommend that you have a look through the paper GAPTrap, a simple expression system for pluripotent stem cells and their derivatives, published in Stem Cell Reports by Cow et al. In 2016. The paper was done under the supervision of Ed Stanley, who is one of the Wells Labs colleagues at the MCRI. After having watched this video, we should be able to understand what a genetic safe harbor is, identify when it may be appropriate to use the GAPTrap system, review how to import plasmids into benchling, identify each part of the GAPTrap plasmids in benchling, understand how each part of the GAPTrap expression system makes it suitable for the system's designed purpose, note the differences between the various plasmids mentioned in the GAPTrap paper, understand how the GAPTrap system integrates its insert into the genome, and understand the basic steps of an experiment that uses GAPTrap. So if you want to insert a piece of DNA into the genome, you can't have it insert anywhere, otherwise you won't know exactly how the gene will behave, and you don't know whether or not it might break something in the cell. Also, you want to make sure that if you are testing different genes inserted into the genome, that each gene is inserted into the same place. So you need to use an expression system that inserts the gene into what is known as a genetic safe harbor. A genetic safe harbor is a site in the genome where you can insert new genetic material where you know that the gene that is inserted will have a predictable function, which means we know exactly how that gene will behave, whether it's turned on or if it's inducible. And we also need to know that that site is a safe site for insertion. That means that inserting the gene won't interrupt any other genes. There are a few known potential candidates for safe harbors. There is AAVS1, CCR5, and ROSA26, which is specific to mice cells. But the one that the GAPTrap system works with is GAPDH, and that's the one we'll be talking about today. So the GAPTrap system is an expression system that is designed specifically for human stem cells. The GAPTrap system works by inserting a DNA sequence of your choice into the human genome using either TALENs or CRISPR, and it inserts this sequence of DNA at the GAPDH safe harbor. GAPDH is a housekeeping gene, which means it is expressed constitutively, or at a high constant level. And GAPDH is one of the enzymes required for glycolysis, which also means that it is expressed ubiquitously in pretty much every cell type. And this is what makes the GAPTrap expression system suitable for use in stem cells. Whatever gene you put in there, you can get consistently high expression, and if you differentiate the stem cell into another cell type, then that expression will continue. So the general plan of an experiment using these constructs is as shown here. So first, you would perform a cloning experiment to put your gene of interest into the GAPTrap plasmid. Then we screen bacteria for the presence of the plasmid using ampicillin. Then you insert the plasmids into your stem cells via electroporation. And then we either use CRISPR or TALENs to insert the plasmid into the genome. We can then select the cells that have successfully taken up the plasmid using an antibiotic, and then you can differentiate these stem cells into something else and see what effects your transgene has. So rather than looking through the figures of the paper, what we're going to do today is look at the same concepts that the figures do, but using benchling. So if we look here, we can see that the paper has cited their use of the plasmid with the add gene accession number 82506. So if we type this into the add gene search box, now we can click on the plasmid, copy the URL, and import it into benchling, like we did in part 1. Clicking on Create, DNA Sequence, Import DNA Sequences, Search External Databases, and then pasting it in here. So here on the right, we can see the map of the plasmid, with each block representing an annotated feature. And on the left, we can see the genetic sequence. So after zooming in a bit, the first feature that I'll point out is EGFP. And by clicking on it, it shows us its location in the sequence. So EGFP is the transgene that the authors have selected for this plasmid. In practice, this could either be replaced with another gene, or a gene could be added onto the end of it so that we have both our transgene and this reporter. The next item that I'll point out is this feature labeled source. So these are the GAPDH tails. These areas are homologous to the GAPDH gene in the human genome. So after we introduce the plasmid into the cell, we can either use CRISPR or TALENs to make a double-stranded break at a specific point at the GAPDH locus in the cell, as well as the homologous point on the plasmid. Then the cell repairs the genome using the linearized plasmid. The authors mentioned that they used both specifically designed TALENs as well as the CRISPR-Cas9 system to create the breaks, and they found that both were highly effective. Now, between the GFP gene and the GAPDH tail at the 5' end is the T2A site. So T2A sites are a type of self-cleaving peptide. When the plasmid is expressed, it translates both genes, the GAPDH and the EGFP, as one long continuous peptide chain. And then afterwards, this T2A sequence, which has been expressed into 18 amino acids, is spontaneously cleaved, leaving two separate peptide chains and leaving our EGFP gene as its own separate peptide. Scientists are still not actually sure exactly how two-way peptides are cleaved, but the current hypothesis is that rather than the peptide chain being produced and then cleaved later, it seems more likely that the ribosome actually skips making the peptide bond between two amino acids in the sequence. So next we have the AMPR gene, which gives a cell ampicillin resistance. This is used in the cloning process when we can select bacterial cells that have the plasmid in it by growing the cells on a medium that contains ampicillin. Now, the authors actually describe a number of different plasmids, 12 of them, that they have created for their paper. So I'm going to have a look at this one here and import it into Benchling. Now, compared to the previous plasmid, this one has two major differences. First of all, they have replaced the GFP gene with a different reporter, mCherry. The authors test 8 different reporter genes in total in their paper to confirm that the expression system is versatile enough to work with many different genes and was not just a fluke. And some of the fluorescent images for each one can be seen in Figure 1 of the paper. The second feature of this plasmid that differs from the previous one is the HYGR antibiotic resistance gene. Now this is in addition to the AMPR gene. While the AMPR gene is used for selecting bacterial cells with the plasmid during cloning, the HYGR gene, which confers resistance to hygromycin, is used for selecting stem cells that have taken up the plasmid. Most of the 12 plasmids that the authors describe have a second antibiotic resistance gene for screening the stem cells. These differences are outlined in Table 1 of the supplementary material, and you can see those differences when you import them into Benchling. And the HYGR gene and the reporter gene mCherry are joined together with this feature, the IRES. An IRES is an internal ribosomal entry site. So when mRNA is transcribed from the plasmid, the ribosome usually binds near the 5' end of the mRNA by recognizing the 5' cap. With an IRES, the ribosome can also bind here, making it a transcript with 2 separate ribosomal entry points. This means that the ribosome can then translate 2 separate peptide chains from the one transcript. Compare this with the T2A site, which creates a transcript that translates into one amino acid chain that is then split into 2. Now, of course, there are other parts of this plasmid, such as various primer binding sites, restriction sites, and promoters, but that should cover most of the important parts. So I encourage you to look through each of the plasmids on Benchling yourself and to look up any other parts that you don't know the purpose of. I hope that explains how the GAPTrap plasmids work and gives you some ideas on how you might want to adapt it for your own project.